and welcome back. With great excitement, I bring to you for free this very special webinar presentation series called I Am Fearfully and Wonderfully Made. It involves 8 amazing principles required for self-healing. These are simple concepts, but it takes a bit more coordination and cooperation to use these tools. We have covered so far 2 of these 8 self-healing requirements. They are pure air and water. My name is Catherine DeRose, your host and health minister, teaching physiological nutrition and its 8 self-healing requirements to help you improve your state of well-being and wellness. This webinar presentation series is made possible by taking excerpts from our Labors with Christ Vagric Manual we taught in Nigeria. Here is the manual, and we are on the topic of our cells. Before we begin our topic today, let's read our primary verse, shall we? I will praise thee, for I am fearfully and wonderfully made. Marvelous are thy works, and that my soul knoweth right well. Psalm 139, verse 14. You may ask, why do we go through this verse every time? Well, primarily, it's a positive thing that we say. First, we're praising the Lord, and we are acknowledging His creativeness in our bodies. And so the more we say it, hopefully the more we will believe it. I would like to invite you to take a trip with me into your body's cells. So we've got a little field trip here today. And I want to tell you some interesting facts you may not already know. And you may be amazed at what you discover. I will share some terms you may or may not have heard, but I will explain about them so you can understand their role and what they are all about. Come along with me. Let's go inside the cell now and take that closer look. Leviticus 17, verse 11 says, the life of the flesh is in the blood. Your blood carries the nutrients to the CBD, or central business district, of our bodies. You can see here our bodies have highways and byways and pathways. Our body is the river of life. It is the blood that carries the essential elements throughout the body, such as transporting that important pure air of oxygen. Let's take a look at the definition of cells. The cell is a fundamental organizational unit of life. All living things are composed of cells, even plants. Blood is the fundamental basic unit of life, full of structure and function in living organisms. Every cell must serve the needs of the whole. And here you have a body full of cells. Our bodies consist of an estimated 125 trillion cells. They come in different sizes and shapes, and they do different jobs. For instance, 1 trillion new cells develop each day, and therefore the same amount must be eliminated. The neurons, the nerve cells in our brains, have star-like branches to network with other neurons. of up to 2 billion mitochondria, the powerhouse that we've already talked about, that generate the cell, each having their own DNA. Mitochondria can power and endanger our cells. Well, how does this happen? Well, our mitochondria make copies of copies of copies, which introduces the possibilities for mistakes. Well, how do they make mistakes? This is possible with the types of inputs we allow. Our mitochondria also component of apoptosis. The cells dying or falling off is another term, as leaves do in the autumn. This is programmed cell death. Self-destructing is a natural part of the regeneration of our cells. Devastating metabolic disorders can result in the next generation.
Mitochondria. Here, food is converted into chemical energy called ATP. ATP is released by the mitochondria so cells can use it. Mitochondria consist of two membranes, an outer membrane separating it from the cytosol, and an inner membrane surrounding the so-called matrix. The area between these membranes is called the intermembrane space. ATP is generated at the inner membrane of mitochondria by an efficient mechanism called oxidative phosphorylation, involving several membrane protein complexes. Nutrients provide high-energy electrons in the form of NADH, which are used by the protein complexes to pump protons from the matrix to the intermembrane space. This continuous pumping creates a proton gradient, where the positively charged protons are attracted to the more negative matrix. When the protons re-enter the matrix through the ATP synthase protein complex, they catalyze the production of ATP.
Every minute, your million cells, 300 million cells in your body die. As our cells die, they are replaced with new cells. 333 billion new cells every day. And the new cell is totally dependent on the building materials that are available.
Necrosis versus apoptosis. Hello everyone. This tutorial will explain to you the main difference between apoptosis and necrosis. Introduction. All cells in a multicellular organism undergo growth and death. Cellular death is essential for an organism to grow and survive. Human body is made up of innumerable types of cells in various parts of the body. There are mainly two types in which a cell undergoes death, either through exposure to harmful environment, injury, or through a pre-planned and regulated process of disintegration. Cell death occurs via two different processes, one being necrosis and the other being apoptosis. Now the question is, what is necrosis? Necrosis is a process of cellular death, which occurs when cells are exposed to extreme conditions, which are very different from normal living conditions. This leads to damage to the internal cellular environment and rapid cell and tissue damage. So the basic inability of the cell to maintain balance when exposed to rough conditions leads to complete discord in the cell's functioning and eventually its death. What is apoptosis? Apoptosis is a pre-planned regular process of cell death occurring in the body where the cell itself actively takes part in its own death. It occurs as part of normal cellular multiplication and turnover. So cells die naturally to maintain a balance of the cellular multiplication. Apoptosis is required for smooth functioning of the body. Most cells in the body have a shelf life. For example, red blood cells live for 120 days. At the end, they are destroyed in the body. This is a method of apoptosis. Difference in cause and presentation. Necrosis is a pathological process which is detrimental to the body. It occurs when the cells are exposed to toxins or on exposure to extreme conditions, which could be anything from increased temperature, decreased oxygen levels, etc. These conditions lead to damage to the wall or membrane of the cell. An inability to maintain normal functioning in these conditions leads to an imbalance in the internal environment of the cell. This leads to inflammation and tissue damage, which leads to accumulation of cellular debris. Apoptosis is a normal, natural process which is necessary for the body, as it maintains a balance in the number of cells in the body. If the cells do not die on time, it can lead to a tumor or cancer formation, which is an accumulation of unwanted cells. If cells die too fast, it can lead to conditions such as AIDS, heart disease, and liver disease. So if apoptosis does not take place as it should, there will be signs of disease. Difference in structural changes. In necrosis, there is a loss of integrity of the wall of the cell, which leads to swelling of the cell's contents as well as disintegration of small bodies of the cell. In apoptosis, there is no disintegration of the cell membrane or wall. Rather, there is a shrinkage of cellular contents along with clumping of the membrane. This leads to formation of condensed bodies known as apoptotic bodies. These bodies are recognized by the immune system of the body and destroyed efficiently by the immune cells. Thus, it is a well-coordinated process due to which there is no accompanying inflammatory reaction. Difference in biochemical reactions. Necrosis is a passive process which does not require energy and can occur at any point of time. This is a random event which is unregulated. Apoptosis is an active process which requires energy and occurs in an organized manner. Many enzymes and agents are required to carry out various steps of this process. Summary. Apoptosis and necrosis are variants of cell death that vary greatly in all ways except the end result. Necrosis is a type of cell death wherein the cell dies an untimely death due to uncontrolled external factors, and apoptosis is a predefined cell suicide wherein the cell actively destroys itself to maintain smooth functioning of the body. Apoptosis is a natural physiological process which is required by the body, while necrosis is a pathological process which stems from inflammation and tissue damage.
Two ways to die, one is clean, the other is chaos. Apoptosis is silent. It's a controlled, programmed death. The cell shrinks, DNA is chopped, and it's removed quietly by immune cells. Necrosis is noisy. It's caused by injury, infection, or toxins. The cell swells, bursts, and spills contents everywhere. This triggers inflammation, pain, redness, and damage to nearby tissue. One is like retiring gracefully. The other is like a sudden explosion. Apoptosis keeps the peace. Necrosis sounds the alarm.
In each cell is a complicated factory, which consists of 10,000 parts like a city. It really is another amazing part of our body. So we're going to travel deeper into our cell and take a look at the city.
The easiest way to understand cell structure and function is to compare and visualize cell with some familiar structures like a factory. It's almost like a mini factory within our bodies. By the end of the video, you will have an interactive understanding of various cell structures and their functions. Let us begin with nucleus. Nucleus is analogous to the headquarters or CEO's office of a factory, where CEO along with managers will decide on products and gives timely instruction for the smooth running of the factory. Inside cell, nucleus is the instruction center, where instruction for the synthesis of products or proteins is coded as DNA. It is protected by a nuclear membrane, ensuring its protection from the rest of the cell. Next is the cytoplasm, which is present in all types of cells. It can be compared to the premise, area, or factory floor where everything is happening. It is the fluid-filled matrix of the cell where all organelles, water, salts, and various organic molecules are present. The ribosomes are similar to the machines in a factory where different products required are made. Ribosomes are protein-synthesizing nanomachines that synthesize proteins required for the cell. Once the product is made, it is to be polished and transported for packaging. Here comes the endoplasmic reticulum, which is analogous to the conveyor belt of a factory through which products are modified and moved for packing. ER is an interconnected membranous system where proteins are modified and transported. Next is the Golgi apparatus, which is similar to the sorting and packaging center of a factory, where products are packed and addressed to reach the correct location. It is the sorting center of the cell, where proteins are chemically tagged or addressed to reach the correct location within a cell or to export outside of the cell. Vesicle is similar to factory trolleys, which transport packaged products to various parts of a factory for shipment. Proteins are packaged in vesicles that are pinched off from the Golgi apparatus and transported to specific locations within the cell or outside of the cell. Next organelle is the lysosome, which is similar to the factory incinerator, where waste materials are disposed and degraded. A lysosome is a membrane-bound cell organelle with digestive enzymes that digest and recycle material taken up from outside the cell and to degrade biomolecules of the cell itself. Next organelle is the mitochondrion, which can be compared to the power plant of the factory, where electric current is generated and supplied to different locations within the factory for the day-to-day activities. Mitochondrion is the powerhouse of the cell, producing energy as ATP by cellular respiration, thus providing energy for all cellular activities. The cell membrane is analogous to the guarded gate of a factory, where credentials are checked before entry. It is a semi-permeable membrane of the cell that has specialized channels for entry and exit of molecules guarded by proteins. It is present in all types of cells, that is in both prokaryotes and eukaryotes. As plants are autotrophs or producers, plant cell has another organelle called chloroplast, involved in the synthesis of food or glucose by photosynthesis. It is just like kitchen or canteen in the factory. In plant cell, apart from cell membrane, there is a rigid outer wall made up of cellulose called cell wall. It's similar to the factory wall, which is a massive wall that surrounds the factory. Finally, in plant cells, instead of lysosome, there is a big central vacuole with all digestive enzymes for biomolecule destruction and recycling, similar to a waste disposal unit or factory incinerator.
Those are the white blood cells rolling around, touching everything. And now we're seeing a close-up of these white blood cells touching. So up at the top is a white blood cell, and you can see these fibers coming down, and then down below is another cell that it's touching. So it's the white blood cell up above, and then it's another cell down below, and they're communicating, but through these fibers, through these touches. So what you're seeing right here is the top of the other cell, and that's its phospholipid bilayer. Remember those balloons with the two strings? That's what this C is. And there's all kinds of things floating on this C, proteins and carbohydrates and all kinds of other things that are floating around. But we're looking at the cell membrane right now. And then we're gonna go inside and look at it. We're inside the cell now, and you're gonna look up. Give it just a second. The camera will look up. We're looking up at the cell membrane, and you can see things moving around in the cell membrane. And now the camera pans in, and you're looking at the inside of the cell. And these, I'm going to turn the volume down. These things that look like pillars and structures, those are part of what's called the cytoskeleton. They're what hold up the cell so that the cell has a certain amount of shape. And you're seeing those things being put together, and you're seeing them being pulled apart. Here, this is called a microtubule. It's what helps the cell to divide, and it's being built. And here's one being deconstructed or pulled apart. Here's everybody's favorite part. This is a motor protein. And I'm going to pause there for just a second. That's a motor protein. It looks like it's alive, like it's a stick figure or something. It's not at all. It's attracted. It's very difficult to explain, but it's attracted to the microtubule that it's walking on, and it's carrying a huge vesicle on its back, which is the products of whatever that cell just made. So if that cell makes hormones, for example, then it's carrying a bag full of hormones on its back, and it's transferring those hormones across the cell. So let's watch him for a moment more. Everybody loves this part. It's super cool. It's like, no way, that can't really be happening, right? Okay, I'm going to pause it again. This is looking still inside of the cell. Way over here, there's a circle, and that's the nucleus. In front of the circle are called centrioles. You don't need to know them, but they're the things that are growing those microtubules that walking guy was walking on. Off to the right is actually a mitochondria, and we'll see that again in just a moment. There's the nucleus more in focus now. This is the nucleus. And do you see that things are shooting out of the nucleus? Those are messages that are coming out of the nucleus. They're called RNA, mRNA, which stands for messenger RNA. And they're going to be captured by an organelle that's called I'm going to turn this down a little bit more. They're captured by an organelle that's called the ribosome. And the ribosome is going to read these messages and interpret them and build proteins. And remember, proteins are what decide what eye color you are, have, or what skin color, or how tall you are, or whatever. And so there's the ribosome grabbing it, the ribosome reads it, and then it builds different molecules, different proteins. Here, a molecule is being grabbed and it's being carried to the mitochondria. That big blob is a mitochondria. Here's another ribosome reading the mRNA and building a protein. That protein is then packaged in a vesicle and it's leaving the smooth endoplasmic reticulum. Here's the walking guy again carrying the vesicle. And now the vesicle is going to join with what's called the Golgi. The Golgi is this enormous layer of different membranes. And that package that walking guy was carrying, it's going to meld with this. And then it's going to be delivered out of the cell. So watch, it's going up. And it's going to be released from the cell. And so whatever was in that package that the walking guy was carrying, it's been released from the cell. Now, if we pause for just a second, we are on the outside of the cell right now. We're sitting on the cell membrane, and still right at the top of us, above us, there's that white blood cell. That white blood cell still wants to know, how are things going? It's checking to see if the cell that we've been inside, if it's healthy. And so now the cell that we've been inside, it's going to tell the white blood cell, hey, we're healthy. They're going to do like a little high five with these proteins that are going to stand up. So it's just going to tell the white blood cell, hey, we're healthy and you can go somewhere else. So watch for the high five. Give it just a second. Okay, they're high-fiving, they're communicating, we're healthy, and now we pan out and we can see the white blood cell, and it's moving on to another cell to see if it's healthy. White blood cells have the ability to change shape so that they can squeeze. That's a real photo. They can squeeze in between cells while they're trying to find if cells are healthy or not.
In this video, we're learning the parts of the cell. Now, I remember first learning this stuff and memorizing each organelle and a little description of what that organelle does, but I never understood the big picture of how these things work together to help the cell accomplish its functions. And that meant that my understanding of the cell and my ability to recall this information, let's say on a test, was pretty limited. So what I want to do in this video is tell the story of protein synthesis, or the making of proteins, as well as a few other cell functions, so that whenever you learn each organelle and what it does, you're going to understand how that all relates together and the different processes that the cell has to do. You're going to understand the cell way better than you ever have. So let's jump to the whiteboard and get started. The first thing I want to talk about is the cell membrane. So our cells are these little bags of fluid, and like any bag, it's got to have some surface around it to keep all of that fluid in, and that's our cell membrane. If we zoom in on the cell membrane, it's going to look something like this. Now, don't click off the video being like, oh my gosh, this looks super complex. The main thing I want to point out with this is that most of the cell membrane is made up of this phospholipid bilayer. In red, we have phosphate molecules, and in the yellow right there, yellow-orange, is going to be lipids. So these are phospholipids, and it's a bilayer. There's 2 layers of them, and this is going to be the membrane that keeps all the fluid in. Now, if you look at this, there's a bunch of other stuff embedded into it, and these are all going to be proteins. So we have a protein here. We've got a channel protein that can allow certain things to pass in and out. Lots of proteins in here, but the main thing, this is a phospholipid bilayer membrane that's keeping all of the stuff in. If this membrane ever ruptures or breaks, all the fluid and all the stuff of the cell would leave, and that cell would no longer be able to carry out its cell functions. It would die. Now, the stuff inside the cell we call cytoplasm. That term cytoplasm, cyto just means cell, and plasm just means fluid. So cytoplasm is the cell fluid. The next organelles we look at, we're going to look specifically at this process of synthesizing or making of proteins. Synthesis means that you take multiple things and put them together in some new form. That's kind of what we do with proteins. We take amino acids and put them together to form a new protein. So let's take a look at some organelles that are involved in this process. The first one we want to look at is the nucleus. So the nucleus, most cells are going to have one nucleus, and the nucleus itself has its own membrane. We call the nuclear membrane or the nuclear envelope. And inside of that nucleus is chromatin. Now you may think of chromosomes. Aren't there chromosomes in the nucleus? Well, most of the cell's life, it's going to be chromatin, which is these kind of long, curvy strands of DNA. It's only when the cell divides that chromatin forms into chromosomes, but most of the time, it's in the form of chromatin. This is your DNA or genetic material, and that's going to be inside the nucleus. So what does that DNA actually do? Well, in the nucleus, the DNA gets transcribed into mRNA. Transcribed just means to take something and make an almost exact copy of it. Now, if you already know about transcription, you know that it's not an exact copy. It's making a copy of the opposite nucleotides, but for the purposes here, just think about it as an exact copy of a section of the DNA. And this mRNA is going to code for a protein. Basically, each mRNA is going to be instructions for one protein molecule. So in the nucleus, we convert DNA into messenger RNA or mRNA. Next, we have something called a ribosome. The ribosomes are super small and they're spread throughout the cell. I've got them drawn in two clusters right here, but they're really kind of spread out more than that in the cell. And what's going to happen is that mRNA is going to travel to the ribosome, and the ribosome is going to read the mRNA instructions and use those instructions to build a protein. Now here's my favorite metaphor for this. It's building Legos. I've got a Lego R2-D2 in the background right here that you may or may not have noticed. But whenever I built that, I had to read instructions, and the instructions told me which bricks to combine in which order in order to make R2-D2. In this metaphor, the mRNA is my instruction booklet telling me what to do. I'm the ribosome because I'm building the protein, which is the finished Lego structure. And each Lego brick is an amino acid. When you combine the amino acids together in the right way, they form a whole protein. And that whole process is taking place in the ribosome. Here is the ribosome. It's reading the mRNA, so the mRNA is feeding through the ribosome right there. And for every 3 nucleotides that it reads on the ribosome, it's going to add in 1 amino acid. So right here, it's going to be building out this long chain of amino acids, forming a protein, each amino acid being one of those kind of orange circles right there. So our created protein at this stage is going to be a long strand of amino acids. Now it's got to undergo some other changes before it's ready to actually do anything in the cell, but that's the point that we're at right now once we're done in the ribosome. So our nucleus contains our genetic information, which is going to get transcribed into RNA in the nucleus. mRNA is going to travel to the ribosome, where our ribosome will read it and then use those instructions to build a protein from amino acids. Now, where do these ribosomes come from? Well, we've got a lot of ribosomes, and we have a special structure that's going to make those ribosomes. And that special structure is the nucleolus. So the nucleolus lives in the nucleus, but its job is to make the ribosomes. It's basically a little ribosome factory. All right, where do our proteins go next after they've been created in the ribosomes? Well, we have another structure here called the rough endoplasmic reticulum. And the rough endoplasmic reticulum is going to be dotted with more ribosomes. All throughout the ER, there are going to be ribosomes, and that's why we call it the rough endoplasmic reticulum. When you look at a cell in the microscope, the rough ER looks rough because it's got all these dots on it, which are the ribosomes. The main thing that the rough endoplasmic reticulum does is it's going to be modifying the protein. At some point, the protein got made in a ribosome, and it passes through each of these layers, and as it does, it's going to get modified by the rough ER. And when we say modified, what do we mean? I've got a list of 4 modifications I want to talk about. One is it'll do the initial folding of the protein. So at this point, we've got this long strand of amino acids, which is our protein, but the actual proteins aren't just a long strand, most of them. They're going to be different kind of 3-dimensional structures. They could be a channel in the membrane that allows stuff to pass through. It could be a receptor on the outside of the cell membrane. It could be an enzyme that binds with a molecule and breaks it apart. It's got to have a 3D structure, and we get that 3D structure by folding the protein. The rough ER can also do quality control, and so if it discovers a protein that's different than it's supposed to be, the rough ER can destroy that protein. It can also do something called glycosylation, which is a fancy term that just means adding a glucose or adding a sugar. That glyco just means sugar. And so a lot of our proteins are going to have other things added onto them. So glycosylation, or the adding of sugar, that'll happen in the rough ER. We can also do destination tagging. So the rough ER can tag the protein molecule with another piece that's going to tell the rest of the cell where this protein needs to go. So is this a protein that needs to end up on the cell membrane? Is this a protein that needs to end up in some other organelle in the cell? Or is this a protein that needs to leave the cell in order to go bind with a structure on some other cell in the body? In other words, is it a hormone? So these are 4 modifications that can happen in the rough ER, but the big thing to remember here is that the rough endoplasmic reticulum is going to be modifying these proteins, getting the proteins ready to go out and do their function. What's next? After it goes through the rough ER, it's going to be packaged and sent to the Golgi apparatus. It's got two sides to it. It has the cis side and the trans side. And that protein, which has been made by the ribosome and then passes through the rough endoplasmic reticulum, it's going to pass through the cis end and then out the trans end. What the Golgi apparatus is going to do is it can do further modifications. So if there's any extra modifications that need to happen before it leaves the cell, it can do that. And it's also going to package it in a vesicle so they can send it to wherever it needs to go. So the Golgi is going to modify and package that protein to send it out to wherever it needs to go. It's kind of the FedEx or the UPS of the cell. Now, where are all the places that it can go? For one, it can be sent to the cell membrane because we have all these membrane proteins. If you remember this diagram from earlier, we've got all these proteins in the cell, like this channel protein that can allow certain molecules to pass in and out of the cell. Another place that the Golgi could send a protein is actually outside of the cell. So it'll package it in a vesicle. We've got an example vesicle right here. The vesicle is going to travel to the membrane and kind of form up with the membrane like this through the process of exocytosis and send those proteins outside of the cell to go do some function. Or it could send them to some other organelle within the cell. Now, there's a lot of different places it could go in the cell. The one I want to talk about, though, is the smooth endoplasmic reticulum. One big difference to remember about the smooth, obviously, is it's not rough, so it doesn't have ribosomes on it. But not only that, its function is very different. Whereas the rough ER was focused on modifying the proteins, the smooth ER is going to be making or synthesizing lipids and carbohydrates. No, wait. We just said we send proteins to the smooth ER. What does that have to do with making sugars and carbohydrates? Well, if you think about proteins, what are proteins? They can do a lot of different functions. One of those functions is to be an enzyme. An enzyme is going to facilitate a chemical reaction. And whenever we're making a new chemical, such as synthesizing a lipid, well, we have to have enzymes to actually facilitate those chemical reactions. So we have special proteins that are made, sent to the smooth ER, so that they can work to make those lipids and carbs that we need. So even though way back at the beginning of this process, we have instructions from the nucleus on how to make different proteins, but really those instructions on making those proteins will also influence what lipids and carbs we're able to make. So it all comes back to that DNA. All right, let's do a quick recap of this process. So the nucleus contains our genetic material, our DNA, which is going to get transcribed from DNA into mRNA. The mRNA will travel to the ribosomes, which are made by the nucleolus. The ribosomes are going to translate the mRNA, read those instructions, and assemble a protein out of amino acids. The protein will travel through the rough endoplasmic reticulum. As it works through those layers, it's going to get modified in different ways. That'll be sent to the Golgi apparatus and get packaged, where it will travel to wherever it needs to go. That could be to the cell membrane, it could be outside of the cell, or it could be to another organelle, such as the smooth endoplasmic reticulum, whose job it is to synthesize lipids and carbohydrates for the body. Next, we're going to look at converting food energy into ATP. Now, this may be the one organelle that you already know from seeing memes online and things like that, and that's going to be the mitochondria. You've probably learned it as the powerhouse of the cell. So the mitochondria are going to be producing energy, but nothing can produce energy out of thin air, right? It can't be created or destroyed. And so we have to be converting it from one form to another. So how do we bring energy into our body? Well, all of this is governed by this reaction. So we have sugar right here. Basically, we've got food that we're bringing into our body, and we have oxygen that we're breathing in. We're going to take both of those chemicals and put them through the process of cellular respiration, and we're going to be converting that into the things that we get rid of, such as carbon dioxide that we breathe out, as well as water. But the big thing right here that we're doing is we're producing ATP. So we're taking these kind of higher energy molecules, we're going to break them apart and then reform them into these lower energy molecules, which will release some energy. And that energy that gets released, we're using that to produce ATP, which is a usable form of energy. Our cells, the pumps and channels and stuff in our cells can use ATP to power all the different functions that we have in the cell. And that whole chemical reaction is taking place in the mitochondria. Fun fact about the mitochondria have their own DNA, which is different than the DNA that is contained inside of your nucleus. And all of your mitochondria you got from your biological mother, none of it from your biological father, which is different than the DNA in your nucleus, which is a combination of your biological father and biological mother's DNA. Mitochondria are pretty wild. All right, let's go on to the next function. That next function is going to be the storage and breaking down of chemicals. We've got 3 organelles that are all kind of similar here. They're going to be involved in these different processes. The first one is a lysosome. That prefix lys, or sometimes light, like cytolytic. Lys just means to cut something open or to break it apart. So a lysosome is going to contain a bunch of enzymes that we can use to break down different molecules that enter the body. This could be a pathogen that's entered into the cell, and the lysosome will spill out its contents in order to break that thing down. It could be an old organelle that we no longer need, that we're going to use the lysosome to break it down. Or sometimes we'll go through the process of apoptosis, which is where a cell decides to destroy itself, and so it'll use its own lysosomes to destroy itself. An example of that would be like an infected cell that doesn't want the infection to spread, so that cell will break itself down. Now, those enzymes in the lysosome, where do those come from? Well, remember, enzymes are proteins. So these were proteins that were made by the ribosomes, which were coded for in the DNA of the nucleus, which then passed through the ER and the Golgi apparatus, and then ended up in the lysosome. So it's even the lysosome, the proteins in there are connected to that process we looked at the beginning of the video. Next, we have something called a peroxisome. A peroxisome is kind of like a lysosome, but instead of breaking things down, it's going to work to oxidize or to neutralize some toxic substances. A big example of that is hydrogen peroxide, which is where it gets its name from. To remember this, just think perox and detox. It'll detoxify certain chemicals that end up in our cells. And then finally, we have the vacuole. The vacuole is just going to be for storage of different chemicals. Plant cells have giant vacuoles that fill up a majority of the space inside the cell. Animal cells, like those in our bodies, have small vacuoles for just small amounts of storage. But certain cells, like fat cells in our body, will have larger vacuoles because they're going to be storing fat molecules. But then a lot of other cells will just have small vacuoles inside. So vacuoles are for storage. The next process we're going to look at is the process of reproduction, which is really going to be either mitosis or meiosis. I say meiosis, some other people say meiosis. If I'm saying it wrong, just let me know in the comments. I know somebody will. Now, I've grayed out a bunch of organelles in the diagram, the ones that aren't quite as involved in this process, but the ones that are heavily involved in the process include the nucleus, of course, because the nucleus has the genetic information, and both copies of the cell during mitosis need a copy of that genetic information. I've got the cell membrane here still because both new cells are going to have each their own cell membrane, and so that cell membrane has to kind of divide and pinch off into 2 new cells. And there's one organelle that we need to add, which is the centrosome. The centrosome looks like this, and it's actually going to be 2 parts to it. It's going to be 2 centrioles. So right here we have one centriole, and then another centriole kind of organized at a 90-degree angle to it. And what the centrosome is going to do is it's going to grab onto the genetic material, the chromatin, which will become chromosomes during mitosis, and pull those apart. So let's take a look real quick at what that process looks like. So here we have the chromatin, and during mitosis, it's going to separate out into chromosomes, which look more like this. And our cell's centrosome is going to make a copy of itself. So we'll have 2 centrosomes now, and those are going to form one at one end and another at the other end. And they'll use these fibers to grab onto the chromosomes and pull them apart so that each new cell is going to have its own copy of the chromosomes. And here we have those 2 new daughter cells that each have a copy of the genetic material. That's how cells make an exact copy of themselves. Of course, we also have this process of meiosis, which is how our bodies create sperm cells and egg cells, each of which are going to have just a half of the chromosomes. But again, the centrosomes will line up on either side of the cell. They're not drawn in that diagram, but they're going to pull the chromosomes apart. And again, involved in that process are the centrosomes, as well as the DNA or the chromatin in the nucleus and the cell membrane. Okay, our next function that we're going to look at is the function of structure and movement. So the first one I want to talk about gives it its structure, and that's the cytoskeleton. Again, cyto just means cell. So this is the skeleton of the cell, and it's made of 2 main parts. It has microtubules as well as filaments. The microtubules look like this. They're these long, thin fibers that are going to originate from the centrosome right here, and they're going to connect all the organelles together. So these organelles aren't just kind of like free floating and wandering around the cell. They're held in place by these microtubules, and those microtubules are made of proteins. So that'll give the cell its main structure, and then on the outside of the cell, we have something called filaments. Now, there's really 2 types of filaments here. There's intermediate filaments, which are going to be more kind of close to the microtubules, and then we'll have microfilaments, which are going to be more on the outside of the cell against the cell membrane. I kind of simplified it here and just called those the filaments. So we have the microtubules holding things in place, and then we have the filaments around the outside of the cell providing some more structure. Let's add a few more things to our cell here. Some cells have structures called microvilli. The microvilli are just going to be these foldings on the cell membrane. The purpose of microvilli is going to be to increase the surface area of a cell. So not all cells have these, but an example of them is the cells in your small intestine. Your small intestine has to absorb molecules that you eat, and so we need a bigger surface area so we can absorb more molecules. So those cells are going to have microvilli. Another structure on the outside of the cell is cilia. The big difference between cilia and microvilli is that they contain microtubules inside of them. At the base of them, there's going to be something called a basal body, which is basically a centriole. And what these microtubules inside the cilia are going to be able to do is allow the cilia to have some movement. So they're going to wave back and forth, so those cilia can move mucus around and move air around and different things. We find cilia in places like our lungs, like the bronchioles in our lungs. One more movement-related structure I want to talk about here is a flagellum. A flagellum is basically the same thing as cilia, but rather than having a bunch of cilia, it's just going to be one projection, but it's also going to be filled with these microtubules. The only human cell that has a flagellum is a sperm cell. And so those microtubules in there are going to have kind of a rotary motion like this, which is going to propel the cell to where it needs to go. All right.
Briefly, we find that each cell has its own biological clock. Take a look at this. And I won't go through all of the stages. It's basically cells replicating and doing what they do.
Keeping our cells young might be a protein called telomerase. This incredible enzyme holds clues to how our bodies age and why we develop diseases like cancer. Telomerase helps maintain telomere length in our cells. These are unique DNA sequences and associated proteins found at the ends of our chromosomes. Each time our cells replicate, their telomeres shorten, like a candle slowly burning down. But when telomeres are way too short, it triggers our body's DNA damage response, signaling cells to stop dividing or die. It's partially through this process that our bodies age. Telomerase is a reverse transcriptase, meaning it uses RNA to synthesize DNA rather than the other way around. Telomerase binds to the telomere overhang and elongates the DNA, then moves down it, repeating this process and extending the overhang even more. After that, another enzyme called DNA polymerase can now attach to the overhang, copying the DNA strand that was elongated by telomerase. If we had enough telomerase in our cells, our telomeres might not shorten at all. But in most of our somatic cells, it's only present in very low or undetectable amounts, not enough to completely prevent the shortening process, but enough to slow it down. This naturally limits how many times a cell can divide, and researchers think it may be a way to protect us against cancer. Unfortunately, this protection is not perfect. Some cells find a way to overcome the limit created by shortening telomeres. Up to 80% of human cancers express abnormally high levels of telomerase. But researchers still have many unanswered questions when it comes to links between telomerase, aging, and health.
What we do know is that if our bodies have too little telomerase, the consequences can be severe. If any of our telomerase or some sheltering genes are not functioning normally, serious conditions like bone marrow failure, where your body cannot make enough blood, can occur.
It's thought that by limiting telomerase, human cells are prevented from unlimited proliferation and the development of cancer. Scientists are still unraveling the links between telomerase and health, but each new study brings us a step closer to understanding how this crucial enzyme might play a role in the future of human health.
This is the point where I want to give our wellness principle that many of these episodes will have. Our wellness principle for this episode is we live and die at cellular level. I'll repeat that. We live and die at cellular level. In the next episode, we will learn more about how important our internal bank is and the importance of making deposits in that bank. Please don't miss out on the next episode. You'll be glad that you tuned in. Now I'd like to close with our closing verse, Deuteronomy 30, verse 19. I have set before you life and death, blessing and cursing. Therefore choose life, that both thou and thy seed may live. Until next time, live your best life now, especially with the improvements. I pray the Lord blesses the improvements that you make. I pray that you are able to choose life and blessing today so that you are able to live free and to the fullest for the glory of the Lord.